Hello, my name is Hala Muhiar, and I am an undergraduate student in Dr. Kevin Shug's lab, and my research focuses on the evaluation of the performance of odor-neutralizing products and components against volatile odorous compounds using headspace gas chromatography vacuum ultraviolet spectroscopy, and this research is in collaboration with Weber Consultants Group. Waste handling operations emit gases consisting of a variety of volatile organic compounds, and many of these compounds have an unpleasant smell, and this becomes a living issue for communities who reside in close proximity to these facilities. By characterizing the odors emitted, we determine odor profiles for the facilities, and based on those profiles, certain formulated odor neutralizers are selected to target their odors. This research focuses on determining which odor neutralizers and single components are most effective by using headspace GCVV. With the VUV detector, we use pseudo-absolute quantitative analysis, where we quantify the number of molecules detected based on a known absorption cross-section of an analyte. This is related back to the amount injected onto the column of the GC. Ultimately, with this method of analysis, we are able to determine a more accurate neutralization value. With headspace sampling, we determine a partition coefficient as another means of assessing how the odor partitions between the neutralizer and the headspace of the vial. In this work, we evaluated various proprietary odor neutralizers provided by Weber Consultants Group and solutions of single components. The single components evaluated include triethylene glycol, tripropylene glycol, polyethylenamine, beta-cyclodextrin, and a mixture of amino acids formulated by Weber Consultants Group. For sample preparation, aqueous solutions of the single components are prepared. A concentrated odor compound standard is also prepared, and there are 3 headspace sample types which are shown here. 1 microliter of the odor compound standard is spiked into each sample type. The water sample type serves as a control, and the empty sample type plays a vital role in the pseudo-absolute quantitative analysis, as I will further explain. These are the instrumentation parameters used. The headspace samples will undergo heating, shaking, and pressurization as ways to assess how the odor partitions between the liquid present in the vial and the headspace of the vial. The Headspace 20 autosampler has a sampling loop that carries a volume of 1 milliliter, which it injects into the GC. But pseudo-absolute quantitative analysis determines how much volume is actually injected into the system based on what is detected by the VUV. The concept of pseudo-absolute quantitative analysis is where the GCVUV can quantify based on a known absorption cross-section of an analyte. This is based on the Beer-Lambert law, which can be rewritten into this form here, where now the cross-section is accounted for. The concept relies on the fact that in chromatographic measurements done with GCVUV, absorbance scans are integrated over wavelength intervals, which converts an absorbance spectrum to a single-valued chromatographic response, like peak sum. Peak sum is obtained by summing the integrated absorbances over a peak. Using this formula here, the number of molecules introduced to the column is calculated and converted to a mass using the compound's molar mass and Avogadro's number. The mass is then divided by the concentration of the compound in the vial to determine the volume injected into the instrumentation. This is where the empty sample type is important because it is a sample type used to determine what volume is injected. This determined volume is used to determine the actual concentration of odor in the headspace of the vial. And so a percent neutralization value can be obtained using this formula here, and a partition coefficient is determined using these 2 formulas here. For the results, Figure 1 and Table 3 visualize and summarize evaluations made on one of Weber's main formulated products and water against common odors. This baseline data for their main product was a starting point to evaluate other formulations and changes that could be made to increase neutralization when it came to client-specific odor profiles. So in Figure 2 is where we performed evaluations on a variety of proprietary formulations on a specific client's emissions. This client emitted mainly aldehydes and sulfur-containing compounds. So formulations V2, 3, and 4 are similar to the formulated product mentioned in Table 3 with small differences, and formulations A through E are different formulations used. For formulations V2 through 4, it is clear that the presence of terpenes does increase neutralization against the compounds evaluated, as V4 had less terpenes and less neutralization. From formulations A through E, it is clear that more amino acids present, which is in formulation D, leads to increased neutralizations. So terpenes and amino acids are interacting strongly with odors. So far with the single component analysis in Figure 3 against select sulfur compounds, it is clear that polyethylenamine and the formulated mixture of amino acids are driving ingredients for neutralization. Thus, amine and imine groups are interacting strongly with the odor compounds and neutralizing their odor and presence. So to conclude, amino acids and polyethylenamine perform well against VOCs that we've detected and worked with, and we will continue to perform the single component analysis and expand the types of VOCs tested. So that is all. Thank you so much for listening.